Today's episode is with Brandon Rust, and he's here to talk about one of the most important topics in your garden, pathogens. You're going to love this episode. Before we get into it, though, quick shout out to AC Infinity. They make the best grow gear in the game: tents, lights, fans, pots, whole grow kits. ACinfinity.com. Use code GROWCAST15 and use code GROWCAST10 on Amazon to save on your AC Infinity products. Thank you to AC Infinity. Let's get into it with Brandon. Thank you for listening and enjoy the show. Hello, podcast listeners. You are now listening to and watching GrowCast. I'm your host, Jordan River, and I want to thank you for tuning in again today. Catch the show now on video. GrowCast.com is where you can find everything. And check out GrowCast.com/membership. Every grower needs a GrowCast membership. We've got the best community in cannabis cultivation. Come and check it out, everybody. Today, we've got a longtime friend of the show back on the program. Our guest is Brandon Rust. You know him, you love him. What's up, Brandon? How's it going, man?
Yeah, what up?
Welcome back to the show, man. How you doing?
I'm good. How are you?
Doing excellent. Listen, man, I appreciate you coming back on the show. There's so many subjects we could cover, but today we have to dive into your recent work researching pathogens. I mean, researching, right? You ran into an issue with some pathogens. You ended up having it tested and analyzed and learned a lot. Can you catch us up right off the bat on this work that you've been doing with leaf spot pathogen research?
Yes. So what was happening was I started to see a kind of a higher pathogen pressure in the area that I'm out in Oklahoma. And I originally thought it was leaf spot septoria. And it has kind of like a limited range of host plants that it can affect. And so it's kind of like plant specific. And there are a bunch of different types of strains of similar microorganisms that are, plant specific pathogens. And so, but what I was seeing was that this particular pathogen seemed to be affecting grass, weeds, the oak around me, just almost kind of everything in general. So I was like, when I started, researching, when I've, learned about the specific strain of septoria, that it's, again, it's really limited on the host species. So I'm like, this can't be that. And I was like, I got to figure out what this is. And so what I ended up doing was I took some leaf samples that had visual symptoms, the spores essentially that form in the center of the lesion that they form. I sent it off to medicinal genomics, and it was actually interesting because I met Kevin McKernan in Puerto Rico at the CanMed, which is like the science conference, and I was telling him about this. I was like, hey, look I'm doing an experiment right now where I have a plant, that I have inoculated with the Trichoderma, the Bacillus, and the Beauveria bassiana, and I have a plant that I haven't. And I know that this pathogen is going to come in. And so what I'd like to see is what this pathogen is. And I, just talk to him about looking at, the plant that shows resistance versus the plant that doesn't. And so I collected a bunch of material, leaves from the cannabis, grass, some weeds, some oak, like a bunch of stuff that I saw visual symptoms of. And they didn't even look at any of that stuff. They only looked at the weed that I submitted so they could identify exactly what it was. And then they sent me the results, right? They showed the whole microbiome makeup, and they did genetic analysis, of what these microbes are. And so we were able to see that it wasn't septoria. It didn't hit on any of their assays. 22% hit for Alternaria, which interestingly enough is just it's an endophyte, and it's not necessarily pathogenic.
So somewhat related to those types of pathogens.
It's just a different strain. It's basically Alternaria, but it would be like if I had, the blueberry train mac crinkle cut, and then I also have the blueberry train mac skunk cut, or it's like I have different phenos of this same strain is essentially how you could look at it.
So that begs the question, Brandon. I mean, do we ever know what we're dealing with when we have something that we recognize as a pathogen? And I will get into with you how to identify a pathogen versus a mineral deficiency. Say, man, it seems to me like we're just literally shooting in the dark. It's like, yeah, it's got a halo. It's probably Septoria. I would have thought the same thing, right? You see the halo, you think Septoria, but when you actually go through the testing, like we don't effing know what our plants
No.
are picking up, do we?
Well, and the thing is, they could potentially be picking up different things at different parts of the season too that look similar. That, here's the thing. So when we look at that test, that Alternaria is the largest dominant species on the fungal report, and then it's followed by several subspecies or different phenotypes, you could say.
Oh.
But different strains of Alternaria. Maybe because there's so much of it as like a dominant microbe, 22% as as fungal percentage, that maybe it's picking up some of that, some like horizontal gene transfer, or it's picking up some type of signaling, or maybe there's some type of nutrient like heavy metals or something that's creating something different in that genetic that's causing the pathogen.
Yeah, the attack.
That's causing the attack on the plant.
Man, that is so fascinating. And this is a problem I see a lot of people, a mistake I see a lot of people make, Brandon, and I'm even guilty of it sometimes, which is when your plant is suffering and you see that you have some damage on your plant leaf, right? How many times do you see people saying, what's this deficiency? And then you take a look at that leaf and what do you see? You see like sometimes black necrotic damage, or like you said, circular damage with halos around them. And where are they concentrated? All around the veins. The veins of the leaf get attacked by these pathogens, where a lot of the time the mineral deficiencies, you'll see that damage literally skips right over the veins. Was that kind of the case with this heavy pathogen pressure? You just saw these scabbing, yellow haloed sores all over your plant's veins primarily?
Well, one of the things about Alternaria is that it spreads its spores, and they're waterborne, they're windborne, and they're really mobile, right? And because they affect a wide variety of different crops, they can move from species to species. They're a necrotic microorganism, or a necrotroph, and so they don't require a live host to sustain themselves, right?
Wow.
So they could live off of dead, decaying material. So it's like if you're composting material and it's not going thermophilic, you're not destroying it, right? So you could potentially be using compost from the year before, adding it into your soil, and then promoting this pathogen. And typically what you'll see is infections from the bottom up. And you'll also see infections bloom really heavily after rain, right? So anytime that where you have overhead rain, and so there's some ways that you can fix it though, right? If you do like, soil irrigation where you're doing like drip lines and stuff, and they're low to the ground and they're not creating any like splashing back up to the lowers. But typically what you'll see is it'll start affecting the bottom and it'll defoliate the plant. And what's happening is there's a bunch of different things happening. So this pathogen is introducing a bunch of enzymes that are basically degrading all of the cellular compartments, so it can basically feed on that nutrition. And in doing so, it starts to defoliate, and so you start to lose photosynthetic capacity. It starts building up and building up, and you can get whole crop loss. I mean,
wow,
it can do a massive amount of damage. And then again, because after it's done with that crop, it can just jump, to another species, or it can exist in the soil in organic material that's decomposing. So it can persist for a long time. And that's why we're seeing an increase in it, is because people have to be more aware that if you're dealing with this, then like you really need to be like taking your material and like getting rid of it, and like not composting it just regularly. If you're composting it, compost it properly, where you're getting it up to like 160 degrees.
Yeah. Quick shout out to Rimrock Analytical, my go-to choice for sex testing, viroid testing, and more. RimrockAnalytical.com, code GROWCAST saves you 10%. Why are you wasting time sexing your plants? You're wasting medium, you're wasting soil and nutrients, and most importantly, you're wasting your time. When it's a little seedling, you can snip off a cotyledon, mail it to Rimrock Analytical, and they will tell you if it's male or female. Again, code GROWCAST, saving you 10%. The best part is the speed. Rimrock Analytical is the fastest sex testing I've ever used, and I see GrowCast members get results on Sundays, getting results late at night. They're working round the clock for you virtually, sex testing your plants. So stop wasting time sexing. Go to rimrockanalytical.com and check out all their other services like HLVD testing and pathogen testing and nutrient testing. It's all there. rimrockanalytical.com, code GROWCAST for savings. Thank you to Rimrock Analytical. That's right. I mean, this is why pathogens are so dangerous. Pathogens like this one, pathogens like Fusarium, is they can hang out in your soil over seasons, like you said, and that can be really, really damaging. That's interesting how you said the soil to foliage transfer. So the splashing of the hand watering, kicking up those spores, and then defoliating, literally devouring your plants from the bottom up. That is very, very interesting, man, and not a great diagnosis, not something you want to have to contend with. How did you fight this infection when you know you had it? I know you use a 1-2 punch. Do you want to talk about that or anything else you used?
Well, it's not just about the 1-2 punch, right? That 1-2 punch being Trichoderma and Bacillus. And those things are amazing biocontrols that have amazing synergy that can suppress a wide variety of pathogens, not just Alternaria. Fusarium, botrytis, powdery mildew, septoria. It has a potential to outcompete pathogens for resources. It has the ability to create secondary metabolites like antifungal and antibacterial compounds, while simultaneously stimulating plant defense systems, upregulating some gene responses, providing phytohormones, and working synergistically with micronutrient interactions to create bioavailability with nutrients that the plants can access. So those are great. Absolutely. I highly recommend them. But it's, proper nutrition is first and foremost, no matter what, right? Proper balanced nutrition, because we're doing some new stuff. We're taking this medicinal genomics thing to the next level, and we're doing what's called ionomics, and we're looking at the holistic picture from an atomic standpoint of what is available and what could be potentially causing a certain organism to do a certain thing. So, for instance, like maybe that Alternaria is completely harmless, but in the presence of some type of heavy metals like chromium, it's just one of the things that we discovered. It has a higher infection rate, right? And so nutrients, and not just plant nutrients, but nutrients that are negative and have negative interactions, can potentially act synergistically with these pathogens. And so it's like, if you have heavy metal contamination, it might be that these pathogens can be more efficient at their jobs because they can strip electrons better using these, heavy metals or whatever.
That is wild. So, it's like the Incredible Hulk. You give it some uranium and now suddenly it's attacking your plant. It's angry. Yeah. What? That's crazy, man. That's really cool research though.
Well, and looking at, so we're doing some projects, and it essentially shows that there are some certain interactions that immediately pop out that, okay, these things are becoming, more pathogenic in the presence of this or in the presence of that. And so we can use these as markers to understand how to steer nutrition in a more efficient way to actually prevent these things from
taking hold.
Yeah.
Let's talk about that. I mean, there are a few minerals that spring to mind that you've talked about on this show, specifically calcium and silica, but do you want to talk about what minerals are going to help us prevent these pathogens from being established so we don't have to knock them down?
Well, it's, definitely calcium, silica are super important when it comes to structural integrity, and then, the the cross linkage that they kind of create intercellularly to strengthen, plant physiology from like abiotic and biotic stresses. It's silica pulp can kind of polymerize into, different compounds inside cells to like strengthen them.
Wow.
And so it can help with plant defense, and it also activates things like the salicylic acid and the jasmonic acid pathways, which are defense pathways that plants naturally and genetically utilize to, defend themselves against invading pathogens and, bacteria. And there's different, mechanisms that the plants utilize for environmental stresses like drought or high salinity or, cold, heat, all of those types of things. And so, silica can help with that. Obviously, adequate calcium, because calcium is responsible for structural integrity. It's responsible for chemical messaging, signaling, and so those are both beneficial things. Silica is not considered an essential nutrient, but it's kind of it's like one of those biostimulants that when you compile other biostimulants like silica and then Trichoderma and Bacillus subtilis and then carbon-based inputs, you're creating massive synergies on so many different levels, from nutrient microbe interactions, micro-plant interactions, plant-nutrient interactions, carbon microbe interactions, root exudates and microbial metabolites, and how they function with carbon substrates and nutrients and, gas and water and hydrogen and all of that stuff, and clay colloids and the surface of organic matter. It's like a crazy puzzle.
And this all goes back to pathogen resistance. So what you're saying is, number one is just having a healthy plant, having a structure that's solid and literally harder to penetrate with things like spores or chewing insects or whatever. And specifically going down into calcium and silica. You mentioned your Bacillus subtilis and Trichoderma combo, right? You also mentioned the kind of removal and defoliation of the worst affected leaves. I think that's often overlooked, Brandon, when it comes to spreading pathogens and keeping pathogens around. And it's one of the reasons I don't love, for instance, chop and drop methods where the leaves are just exposed. That worries me, man. I've had too many pathogens. I see a lot of people do it with success. So, good on you, Godspeed. But I'm not doing that because I'm too worried about pathogens, Brandon.
Well, here's the thing. It's always good to do your due diligence, because even if you don't have a problem, it doesn't mean that you can't get one. And if you always take proper procedures so that you don't, then you don't
have to worry.
Yeah, exactly. And so, and it's not so much, you just need a healthy plant, because healthy plant starts with healthy nutrition, right? Regardless of how you grow. Because, obviously I consider myself an expert in, living soil systems, agronomy, and microbiology. And for me, that's my main focus. But, I also did hydroponics for 12 years consistently, outdoor and native soil with, organic, and I've done it inorganic. I've done every kind of grow method, and what it really comes down to is it all starts with nutrition, man.
That's a really good
proper
point.
Balanced nutrition. So if we're talking about soil, what that really means and what that equates to is, the amount of carbon in your soil functions as an energy source and a battery to power microbial metabolism, to power things like the carbon and nitrogen and phosphorus and micronutrient cycling processes that, whether, the lithosphere, the inorganic mineral base, and convert it into plant available cations and anionic nutrients.
I barely followed you there, but I absolutely got you. And on that note, viewers, if you want to learn more about this pathogen study, if you want to learn more about carbon-based nutrients, at Full Crop Sciences is the new Instagram. We lost the old Instagram, Brandon. That's too bad. I heard you're fighting to get it back. Good on you.
Yes.
The website is bokashiearthworks.net. biocontrols.net. The code is GROWCAST. Members get an extra special code. And the Instagram is at Full Crop Sciences. That's FUL, Full Crop Sciences. So go and give a follow. And if you're checking out the carbon-based fertilizers, it's code GROWCAST at bokashiearthworks.net. So you can grab the Bacillus subtilis, the Trichoderma, and you can grab the Calcil, loading up my earth boxes with calcium and silica, front loaded with that Calcil. I love it, Brandon. But on the subject of that 1-2 punch and pathogens, Bacillus subtilis and Trichoderma, you named a whole bunch of benefits that they bring to your soil. Why specifically are they so good for fighting off pathogens? What about these two, or each of them, or both of them together, works so well?
So Trichoderma has been shown in so many scientific studies to just be such an aggressive fungal species, to where it can essentially utilize any type of resources around it much more efficiently. And the way that it does that is it produces secondary metabolites. You can think about these metabolites similar to terpenes, right? Different terpenes or different cannabinoids, they cause different effects, right? And so the same thing can be said with these secondary metabolites that these microbes produce. They create biochemical reactions in soil with nutrients or with the plant.
Mm-hmm.
With the trichoderma is really interesting because trichoderma works just like mycorrhizal fungi. So it can create plant root associations and create fungal hyphal networks that can transport nutrients and water and minerals and everything the plant needs. And that also increases essentially the root biomass. And anytime you have a increase in the percentage of biomass or the surface area of biomass, you have the more potential for reactions to happen. So that's one of the rules of like thermodynamics. You know, the larger the surface area, the greater the reaction, right? It's just like why we micronize things down, because they can become more soluble or because they'll react better.
Right.
And so the trichoderma not only works as a mycorrhizal fungus, but it actually becomes an endophytic fungus as well. So what it'll do is it actually will colonize the vascular transport mechanisms of the plant. And so it'll live inside of the xylem and it'll actually procreate and grow. And the only way that it could possibly do that is by using the solutes that the plant are actually taking up. And so what it's doing is it's using some of the nutrients that the plants are using, and then in exchange, and probably using carbon substrates that are being taken in. And who knows? That's where the science is kind of unknown.
Wow.
I would guess and hypothesize that just like the metabolites that are released into the soil, things like phosphatases, amylases, phytohormones, any of those beneficial compounds that those microorganisms naturally produce through secondary metabolites or through secondary metabolism would be produced internally in the plant. And so what exactly is happening? You know, are those chemical compounds acting as chemicals, carbon skeletons for different enzymes and proteins and sugars, or are they being catabolized for energy? So that way they're, we just kind of, at this point, we don't know. And that's kind of what we want to find out is with the ionomic research, is to find out what is happening when, these microbes are interacting with plants like this. And so and then Bacillus works synergistically, because the Bacillus itself as a bacteria is much smaller and it can colonize the fungal filaments or the hyphae that those Trichoderma make, whether they're, acting as a free living soil microorganism or whether that Trichoderma is attached to a root and acting like a mycorrhizal fungus. It can colonize that. And they're producing really similar metabolites that actually make each microorganism function better together. And that's why we call them a synergistic microbial consortium, is because you're getting dual efficacy because you're coming at it from two different angles, a bacterial angle and a fungal angle, and they both work together to reduce fungal pathogen resistance, to outcompete pathogens, to cycle nutrients, to create, phytohormones that stimulate, plant responses, and the magic of microbes, man.
I think it's one of the most effective combos because there's a lot of stuff out there, but these two things are really the powerhouse behind your garden and your soil, Brandon. It's really cool how they synergize so well with each other. And like you said, how they provide both beneficial, reactions and interactions with the plant, and then also so good at the IPM. I think depriving the food source of the pathogens is a big thing that I've kind of overlooked because I know Trichoderma, releases compounds to kill pathogens and even digests them and grows around them. But the fastest way to get rid of anything is to take away its food source. And that makes a lot of sense to me. So I think that's one of the big takeaways for this episode, listener, is front load that good nutrition, specifically your calcium and your silica. Make sure you're taking good care of your foliage and not leaving around dead material that could house this type of pathogen that also feeds on living and dead material. And then, of course, front load the microbes too. Make sure you've got tons of microbes in there. I love the 1-2 combination. I love this type of education. Always dropping knowledge bombs on the show, Brandon. I want to make sure that we have time to send listeners your way, though. Can you talk about anything you got going on with Bokashi Earthworks right now? Anything you want to tease? Of course, BokashiEarthworks.net, code GROWCAST. But what's going on in your world?
So we're trying to build out our command center. We're going to be focusing a lot on email marketing and letting people know when we're doing podcast drops, giving out guides to different things that can be helpful for people. And
so go sign up for the newsletter right now. It's free. BokashiEarthworks.net.
And so look, like right now I did a free guide for dry cure that's available. That is, if you go to the blog on the Bokashi website, that guide is free. You just go to the blog and you can copy paste whatever, or you can download it. I think also, whether it's, blogs or guides, or whether it's the Discord channel, or whether it's the YouTube, or just the social media, or whether it's signing up for our newsletter and making sure that they're getting all the coupon codes when, throughout the year, whenever we're having sales. It's just, trying to put everything together in a really functional ecosystem. So that way everybody feels included and appreciated.
That's awesome, man. We appreciate the code GROWCAST 10% off. We appreciate the member code. They're getting 15% off in membership only. So you're a great friend of GROWCAST, Brandon. If there's anything we can do for you, let us know. Go follow at Full Crop Sciences, at Full Crop Sciences. Go follow now. And we wish you the best of luck, Brandon. Hey, keep us up to date on that research. I want to see what you guys find as you dig into the different relationships.
Oh, yeah. It's going to be interesting. Look, we're working with OSU, the professor of biology at OSU, working with Cove Environmental. So we have a solid research team of PhD scientists. And of course, I don't have any, educational, technical degrees or anything like that, but I've got 25 years of professional cannabis cultivation experience, and over a decade of, science-backed, data-driven, cultivation and research. So
that's awesome, man. Find it at BokashiEarthworks.net, everybody. Code GROWCAST. Thank you, Brandon. We appreciate you. Thank you, listeners, viewers. We appreciate you. GrowCast.com is the website. Go and check it out. Everything's there. The seeds, the membership. It's all waiting for you. And until next time, this is Brandon Rust and Jordan River saying, be safe out there, everybody, and stick around with GrowCast so you never have to grow alone. Bye-bye. Peace. That's our show. Thank you to Brandon Rust. Thank you so much for tuning in, and stay tuned. GrowCast.com for all your GrowCast needs. Check out GrowCast membership. Every grower needs one. Not only will we support you in your garden 24/7, but we've got an awesome community of growers. We do grow-alongs at the Grand Pheno Hunt where we're giving away $1,000. We have growers connecting in the trading bazaar and meeting for meetups. It's truly the greatest community in cannabis cultivation. Come and check it out. GrowCast.com/membership. But that's all for now, everybody. Stay tuned. Hit like, hit subscribe if you learned something from this episode. I know you did, and I will be back soon with some more great content just for you. All right, everybody. See you next time. Bye-bye.